Okay, if everybody can get seated, because we are preparing for a very interesting second keynote presentation given by César Marín, and he is full professor at the Universidad Santo Tomás in Chile, and he is very active. If you look at his CV, he is editor of various journals. He is organizing networks. He is vice chair of NetSop. And if you look at his papers, he has published about an interesting variety of aspects. Sometimes it gets pretty philosophical, and this is what I personally also very much like. In his announced title, it was also something initially about philosophy. So let's give the floor to César. Welcome. And give a big applause to him.
Thank you very much for that nice introduction, and thank you to the organizers, to the GSBI. It's a true honor to be here this afternoon. I have known about the GSBI since I started doing soil biodiversity research, so this is very neat. Using complexity science and philosophy to assess mycorrhizal cooperation, I think that I will deliver more on the philosophical part, and the complexity part is kind of a dream, or is kind of the next step. So this talk has these 5 components. First, we will talk about context dependency in mycorrhizal functioning, then about natural selection at multiple levels, then about a brand new framework that me and Nancy Collins-Johnson call functional team selection, some few slides about how is to do soil biodiversity research from the Global South, and some concluding remarks. So I think that one of the best known aspects in mycorrhizal functioning is context dependency. In particular, the continuum to mutualism parasitism, the continuum mutualism to parasitism continuum, in which fungi are always benefited, while the responses for plants could be mutualistic, could be neutral, or could be even parasitic under some restricted conditions. One important aspect of this article, which already has almost 30 years, is that the mycorrhizal phenotype is affected by the plant and fungal genotypes, and also the rhizosphere and community and ecosystem characteristics. And from that point on, we can understand the mycorrhizal phenotype as an interaction of all of these factors. I think that in our community of mycorrhizal ecology, people kind of have forgotten about this, about the fact that it's an interaction. And well, among the first aspects that we understood is that nutrient limitation increases the degree of mutualism. So we have evidence from meta-analysis like this one, like this one that's already 15 years old, in which we know that mycorrhizal growth response is higher when either nitrogen or phosphorus are limited. From the metabolic point of view, we also know that a high phosphorus content almost completely suppresses the metabolic absorption pathways by mycorrhizas. And this 2023 paper by Ma and co-authors shows that global arbuscular mycorrhizal fungal diversity and abundance decreases with soil available phosphorus. So that's also a very strong kind of ecological evidence of this mutualism increased under nutrient limitation. In my own research in Chile, we also know this pretty well. I know that doing taxonomy through spores is very time consuming and very old fashioned, but I think it's very important to keep doing it. So we have sample spores across 2,000 or 3,000 kilometers, mainly in forest ecosystems. And in this publication last year, we got to a total of 71 species in the country, and 803 occurrences. And yes, it's definitely the case that in forests with very low phosphorus, I'm talking about 3 parts per million of Olsen phosphorus, there is really high diversity of AMF. And in contrast, forests with 50 parts per million or crops with 50 parts per million will have only 2 or 1 species. It's not only about nutrients, though. We also know that some abiotic stress increases the degree of mutualism. Here, 2 collaborators, Paula Aguilera from Patagonia and Geomar Vallejos Torres from the Amazon in Peru. We have published several articles showing that in the case of Patagonia, these mycorrhizas are very beneficial, particularly on the high aluminum concentrations in the soil, which these volcanic soils naturally have. And in the Amazon, very high cadmium concentrations lead to an increase in this degree of mutualism, of mycorrhizal mutualism. So, of course, this has important consequences in agricultural applications. This research, this study by the Van der Heyden group in Switzerland, I really like this paper. It's one of the largest in situ mycorrhizal studies, 54 cornfields in Switzerland. If you see, only in 25% of these sites, a mycorrhizal growth response was higher. I mean, the inoculated treatments were higher. Then in about 50%, they were the same than the control. And in 25%, they were lower. Before the inoculation, they measured 53 predictor variables, and they found that the best predictor was when soils had a high initial abundance of plant pathogenic fungi, which by the end of the trial has drastically decreased. So it's also about biotic stress. Nancy Johnson in Northern Arizona University has conducted these 3 or I think more really neat experiments with sympatric and allopatric pairings of initially just AMF, but in later studies it was whole soil microbiome. And the main conclusions of these studies is that when plant host mycorrhizal fungi and the soil microbiome come from the same origin, meaning that they are sympatric, mutualism is enhanced. Plants grow more, they tolerate stress better, and fungi also grow better. So they measure things like extraradical mycelium and spore production, and this sympatric combination was also beneficial for fungi. We did a sort of similar experiment with Araucaria araucana, which is the Chilean national tree. It's a conifer. Conifers in the southern hemisphere, they are arbuscular mycorrhizal. It's grow very slowly. So first we had this experiment in a greenhouse for 1.5 years, and basically we had 4 treatments, a control, 2 commercial products in blue and green, and then native AMF in purple. As you see, the growth was higher with the native AMF, I think in part because the native inoculum consisted of way more species than the commercial inoculum. But from this experiment in the greenhouse, we took the control and then the native inoculum treatment, the cuttings, like maybe 15 centimeter cuttings, and we put them back in the same forest in which the seeds were obtained, which is this graph. So, and then in a contrasting nearby forest. The other big species in Chile is Notophagus, which is ectomycorrhizal, and will often grow right next to the Araucaria forest. So we put these cuttings in the own forest, in Araucaria forest, and then 300 meters apart in Notophagus forest, which is ectomycorrhizal, which is a forest with more light, more water, but still these Araucarias grew better in their own forest of origin. So that's very intriguing from my point of view. So far we know that mycorrhizal mutualism increases under nutrient limitation, increases under abiotic stress, biotic stress, as the Van der Heyden studies show. So mycorrhizal mutualism increases when we have limited or stressful conditions, or both. And these studies by Nancy also show that sympatric conditions are highly, highly important in increasing mycorrhizal mutualism. So, multi-level selection theory. This has been also a big passion of mine for the last 12 years alongside mycorrhizal ecology, but they were always like two frameworks of work which I'm kind of now finally putting both together. To start, this couple of papers from the last 2 years in which I argue, and along with Mike Wade from Indiana University, we argue that a gene-centered view of evolution has very unrealistic assumptions. First, that populations are huge, unstructured, and the interactions between genes, individuals, and environments are totally random. Also, this gene-centric view of evolution does not capture phenomena that we now know that are quite common, like epistatic interactions, are widely common across all taxa. Indirect genetic effects, which means that in epistasis, your genes or the genes of an individual interact in a very complex way to explain a network of traits. Indirect genetic effects mean that genes in the population also interact in a very complex way to affect the phenotype and the fitness of a focal individual. And this is also very common. A gene-centered view of evolution doesn't capture non-genetic inheritance, epigenetics, and so on, horizontal gene transfer, niche construction, and multi-level selection. So what we argue in this article is to simply bring back the phenotype, to put attention back in the phenotype. And I think that is very telling on how our different field, particularly microbial ecology, has evolved over the last 30 years, in which at some point we all were doing metabarcoding, then proteomics, then metabolomics, and now I think that the next big thing is trade ecology, and a lot of people are doing trade ecology, rightfully so, because we have seen that all these previous frameworks were not enough to explain the phenomena that we are trying to describe. As a reminder, what is natural selection? I think one of the most real examples of natural selection is antibiotic resistance. So we start with some resistant bacteria. We apply the antibiotics, and then because of their enzymatic activity and some other traits, those resistant bacteria become more abundant quite quickly. This is a daily example of natural selection. What Richard Lewontin described in 1970 was the 3 requirements for any biological entity to evolve through natural selection. So we have phenotypic variation, differential proliferation, and inheritance. In the bacteria, we have some resistant bacteria, some non-resistant bacteria. Then the resistant bacteria proliferate more, and this resistance is inherited through several generations. So Lewontin's proposal with this paper was that we need to evaluate these three things across many different entities. This is kind of an abstract characterization of what natural selection is. Then, more or less in 1985, a brilliant philosopher of biology, Lisa Lloyd from Indiana University, she analyzed this framework and she found that what happens in natural selection is that we have three different functional roles. One is the interaction, one is the replication, and one is the manifestation of adaptations. So she argues that an interactor is a biological entity that interacts through its characteristic with the environment in such a way that proliferation is differential. When I first started to kind of really dig into the philosophy of biology, the terminology is sometimes confusing, I need to be honest. But the best way that we can think about this, organisms have traits, they reproduce in a differential way because of these traits, and the consequence of this is that genes are replicated in a differential way. That's the best way that we can understand about this. But what is interacting with the environment are the traits and are the phenotypes, not directly the genes. And that's a crucial point to keep in mind. And the first kind of unit of inheritance for a long time was the replicator, the genes, but then again in the 80s and 90s we started to discover epigenetic inheritance and cytoplasmatic inheritance and RNA inheritance and in social animals, cultural inheritance. So we don't need to think about inheritance only as genes. Many things get inherited through several generations. So what is multi-level selection? Has been defined as a situation in which natural selection occurs among entities at two or more different levels in a nested biological hierarchy. So when we have natural selection in the individuals, in groups, in communities, or that's natural selection above the individual level, we can also have natural selection at the cellular level, and the best example of natural selection at the cellular level is cancer. And there is a whole group in Arizona State University doing research in cancer through this framework. How do we measure multi-level selection? There are two ways. We can either measure differential reproduction of entire groups, which is quite challenging, or when we have differential reproduction of individuals based on their group composition or characteristics. In that scenario, we also have multi-level selection, because it's a simple equation I will show later, which partitions natural selection at the individual and the group level. I think this is a very hot topic in evolutionary biology since the 1960s. There were very famous books arguing from one position and arguing for the other position. So The Selfish Gene is a book that argues for the gene-centric view of evolution, but then Lewontin and then Sociobiology by E.O. Wilson is kind of in the middle. And I think part of the confusion is that one focus has been the evolution of altruism. Altruism is one of the traits that could evolve through natural selection and through multi-level selection, but it's not the only one. But I think big part of the debate has been because of altruism. In any case, of course altruism is important, and of course cooperation is important. And in this paper by E.O. Wilson and David Sloan Wilson from 2007, they explain the evolution of altruism through multi-level selection in the following way. They recognize that at the individual level, definitely selfish individuals have an advantage over altruistic or prosocial individuals. There is no doubt about that. But at the group level, groups composed by more altruistic or prosocial individuals will have an advantage over groups composed by more selfish individuals. This was said by Darwin in The Origin of Man when discussing moral behaviors in humans. He said something like, a tribe composed by highly moral and altruistic individuals will outcompete tribes composed by more selfish individuals, and this will be natural selection. So at the end of the day, there is this balance between forces at different levels of a nested biological hierarchy. And I think also a big part of the debate in the 60s was a book by a biologist, Williams, in which there was a naive version of group selection. So Williams back then argued that all behaviors, he was a social behaviorist, all behaviors in some birds, I think, were for the good of the group. And that's, of course, a naive version of group selection or natural or multi-level selection. This framework has been applied in many disciplines, in molecular biology to explain the central dogma, in cultural evolution, in economy, and to explain the COVID. And I will talk about two of these examples in the slide in a few minutes. So this year in February, we published this article, which I'm the only kind of a mycologist or microbiologist there. All my other co-authors are zoologists, and it's always very interesting to cross this line and work with people outside your expertise. And in particular, Mike Wade is a fantastic quantitative geneticist from Indiana University, who is one of the pioneers of empirically studying multi-level selection in Tribolium, which is a beetle, a flower beetle. So his PhD thesis, published in 1976, was kind of the first experimental evidence of this in PNAs. So we did a bibliometric analysis. We went through a bunch of almost 3,000 articles, and we defined criteria of selection, and at the end, we have 280 articles providing empirical evidence of multi-level selection. As I said before, the first one is by Mike Wade in 1976 in PNAs, where he imposed group and individual selection treatments in these flower populations for many generations. And for a long time, that was most of the research with animal populations in laboratory conditions, altering different conditions of the treatments, but then research in situ came, and since 2012, more or less, there is quite significant amount of research. So we dissected the levels of selection that were investigated. 90% of the studies were focused on organisms and groups of organisms. So 90% of the studies detected natural selection at these two levels, organisms and groups of organisms. Because that's the focus, I think organismal biology is the focus of most of us that do biology. So that's perfectly reasonable. So 24 studies focus on aggregates, and by aggregates I mean bacterial colonies or colonies of aggregates of amoeba, algae, yeast, and so on. And 31 studies were at the colony level, particularly eusocial insects like ants and wasps and bees. But then almost 10% of the studies were above or below the organismal level. Studies at the nuclei level with arbuscular mycorrhizal fungi, actually, the cell level, genomic element level, module, and so on. So I just want to highlight 3 of the most striking examples. Give me 1 second. So one area of research in which is very evident, in which multi-level selection is very evident, is these artificial selection experiments in which they particularly use yeast. If I remember correctly, there are like 12 studies of this type in which they basically try to explain how multicellularity, what was the origin of multicellularity, and these yeasts are under different treatments like anaerobic and obligately aerobic and mixotrophic. This one in Nature from 2023 is one of the best ones, is really striking. After 600 rounds of selection, in the anaerobic treatment, these groups of yeast evolved to be macroscopic, many times larger, more tougher, and they retain a clonal multicellular life cycle. So this is one of the examples that I really like. Then people doing animal breeding also use the logic of group selection and multi-level selection for a long time, particularly Peter Bigma from Banningen. In these breeding experiments, you can have a control and you can have the typical breeding program, which will select the hen that produces more eggs, and then the cage that produces more hens as a cage, not as an individual hen, but a cage with many hens. So they bred these hens, these chickens, for many generations, 30 generations, 40 generations, and the result is that the group selected lineages are way better, produce more eggs, and more importantly, are less aggressive between them. These group selected lines drastically reduce cannibalism and aggressiveness in the hens, and this is now a standard practice in many fields. This kind of studies were 32 studies. One way to measure multi-level selection is using this tool, which comes from quantitative genetics. It's a very simple multiple regression, contextual analysis, in which the individual relative fitness is a function of individual, group, and contextual traits. So I did this myself. This agronomic experiment in Colombia for 2 years, in which the seed biomass of these corns were a function of the traits of the plant, plant height, stem diameter, shoot biomass, etc. But also in the same regression, I included the average traits of the neighbors. So in that way, and then contextual traits like density and spatial pattern, which only can be measured at the group level. So in that way, we can partition natural selection on individual and group level components. So I did one of those studies, but there are 26 of these studies in total. The first one from 1995 up to last year. Connor Filson, he followed a marmot population in California for 19 years, doing this kind of analysis. And in this graph, I show the effect of organism size on individual relative fitness at the individual and group level. So the green dots represent the regression coefficient of the individual level, and then the purple of the group level. And as you can see, in some cases, individual and group level go in the same direction, maybe like here, and sometimes they go in opposite directions. And sometimes it's stronger, the coefficient at the individual level, and sometimes it's stronger at the group level. The main conclusion for this is that we cannot generalize. We need to measure these things in real time, in situ, and that also sometimes individual and group level go in the same direction, and sometimes they go in opposite directions. And I think one of the most striking examples of multi-level selection is by Elinor Ostrom. She was the first woman to get a Nobel Prize in Economics in 2009, not surprisingly. This paper is with one of the architects of multi-level selection theory, who is David Sloan Wilson. She went out and studied hundreds of human groups all around the 5 continents to see if these human groups reach the tragedy of the commons, which is a very well-known concept in ecology and economy, or if they are very aware of this, which is the case, and they don't get into the tragedy of the commons. The answer is that no. Human groups are very aware of this principle, so they have developed these kind of universal principles to avoid sobre-exploitation, to avoid this tragedy of the commons. Sometimes these principles are laws, sometimes they are tales, sometimes they are in the form of religious principle, sometimes they are myths, but she analyzed these principles across all these human populations, which is intrinsically a multi-level selection framework. So, I said before that one of the objectives of this talk is to check how context dependency on mycorrhizal functioning and the reality of multi-level selection could be combined, which is functional team selection. So I have been thinking of this for a long time, but one of the triggers to come to this framework was the very well-done research on the ionosphere microbiome. There is very neat research in the Czech Republic, in China, in different places, which shows that basically we need to stop thinking about mycorrhizas as a 2-actor interactions, and we really need to incorporate the microbiome in the IFE. So, for example, this paper by Letian Wang and co-authors, they found ionosphere bacteria that are able to mineralize phosphorus, and that there is a core functional microbiome in very different ecosystems, corn ecosystems in China, and they found the same core microbiome in microcosm experiments. What I like about this paper is that they use module analysis to define their core microbiome, because it's not that the taxa coincides, the specific taxa coincides across all these different ecosystems, but rather the functional role of this taxa. And then this other study by Zhang et al., they manipulate, they separate the ionosphere from the fungus and the plant, and they have all possible combinations, a very well-designed experiment, and they conclude that arbuscular mycorrhiza do not function in isolation of this ionosphere microbiome or ionosphere bacteria. So we argue that arbuscular mycorrhizae, the IFE, is an enormous but unexplored ecosystem in which evolutionary process take part all the time, probably at the scales that we are not still measuring. And the other kind of trigger of functional team selection was to understand these nested levels of biological organizations that we have, and I'm sorry for my bias, which is arbuscular mycorrhizal fungi, but I think that the same could be said about other mycorrhizal types, but in particular AMF are very interesting because we have, of course, genes and transposable elements in the genes, and then we have the spores, and you know that if you don't know, the spores can have 4,000 nuclei, and sometimes the nuclei are identical, but sometimes they are different from the genomic structure is different. And this is not in the figure, but there is also very good research by Olga Latovetsky from Ireland showing bacteria inside the spores, which are still very understudied. We still don't know a lot what they do. And then, of course, we have the spores, and we have the rhizosphere and the plant holobiome in general. So in this paper, they invite to think about these different levels of, it's kind of a Russian doll thing, to try to understand how natural selection could operate across all these levels, but also try to think what are the roles of these different entities of the spores, the spore traits, the holobiomes, the genes as replicators, and so on. So, knowing that mutualism increases under limited or stressful conditions, knowing the evidence of local adaptation, in particular that the sympatric pairing experiments, knowing that there is abundant empirical evidence of multi-level selection, the hyphosphere role and units of selection in mycorrhizas, with all these ingredients is that we came to functional team selection. This is the paper of last year in ISME journal. And one important aspect is that we, in this paper, define the holobiome as a complex adaptive system. Ever since the appearance of the holobiome or hologenome concepts from 2007, there are at least 6 or 7 different concepts of holobiome, and each concept is based on different disciplines. So there is a genomic definition, there is a population genetics definition, there is a philosophical definition, and we didn't want this has been debated a lot, so we didn't want to go into the weeds of that debate, but rather, if you see the characteristics of what a complex adaptive system is, I will not read all of them, they are perfectly compatible with a plant holobiome in particular. And if you put attention to 6, these complex adaptive systems generate emergent properties in which the whole is different than the sum of the parts. And I think this is crucial. So plant phenotypes are the sum of all these different complex interactions. And of course, here we include eukaryotes and prokaryotes and viruses. So there are 2 important concepts in this framework. One is a functional team, I will go over later on, but the other very important one is a positive plant soil feedback. So for a functional team to assemble, we need these 4 things to happen. We need a selective force. So if there is a limiting resource or a biotic or biotic stress that can be ameliorated by the host microbiome interaction, we need host constitutions. We need plant hosts that are able to recruit these different microbiomes through incentives and deterrents. And this is important because I think microbiome recruitment has a phylogenetic signal, so not all plants are able to recruit different microbiomes. Then, is there enough microbial diversity in the system? Some soils, I have done research in some soils, very poor soils. So, and the 4th one is time. Is there sufficient time to assemble these functional teams? So if the answer to these 4 questions is yes, we predict a functional team, and a positive plant soil feedback is probable or is likely. This commentary paper by John Klinoromos and co-authors shows this in another graphical way, in which limited resources predict a functional team and a positive plant soil feedback. And if we increase resources, there is either no team or even a dysfunctional team. So in a way, coming back to the 1997 paper, in which Nancy Johnson and co-authors talk about commensalism and parasitism and mutualism, 2 actors interactions. Here we are talking about trillion actor interactions. So one question is how much time, and this is still, I'm still thinking about how much time does it take to assemble a functional team, because the lifespans of all these different actors is so different. So some plants can live thousands of years, and clonal fungi can also live significantly, but then bacteria, in a day you have 3 generations of bacteria. So time is an open question. Some recent work of us is intriguing. I'm not saying that this work proves that time is important, but at least these results are intriguing. In this study in Chile, in 40 vineyards, we had vineyards of 1 year old and vineyards up to 41-year-olds. And we definitely saw that maturity increases arbuscular mycorrhizal fungi and then decreases significantly plant pathogenic fungi. So that's intriguing. But of course, that's only one of the factors in our framework. So it's only time. We need to test all the other factors. This other recent study in collaboration with SPUN, I'm very lucky to live really close to this magnificent conifer, Fitzroya cupressoides or Alerce. Is a tree that can live very, very long time. So the individual that you see in the photo is called the abuelo. And the 40% of the tree, because the tree is so huge, the dendrochronological tool only reaches 40% of the tree, and that 40% is 2,400 years. To estimate the real age of the tree is next to impossible, but at least we can say that it's minimum 2,400 years. And in this study, we had 31 trees, including this one. The youngest one was like 25 years, and then different ages. And then we kind of see the same thing. We see that, in particular, AMF increases with tree size and tree age. So, some few words about doing soil biodiversity research from the Global South, which is something that I have been focused over the last 12 years. There is this colleague, philosopher, Laura Nuño de la Rosa, and she's researching how the backgrounds, the cultural and ideological and socioeconomic backgrounds of scientists kind of put the type of research that we do. I think that there is an obvious influence on how we were raised, how the context that we grew up. I grew as a coffee farmer in Colombia. And yeah, that gives you a very different perspective on how to do science and what do you care and what you don't care about when doing science. And also when I know that we in the academic world have a lot of problems, a lot of injustice, and a lot of underfunding and so on. But when I go back there to my family, this feels like a total privilege. It's a complete privilege to do this. So I have been thinking about this couple of papers also this year. In one, we discussed the inequalities of doing research based on language and funding barriers. So one of the authors from Iran, he mentions that if he wants to pay a Nature APC, that's like 80% of his budget for a year, so it's next to impossible. And also about language, we discussed that we need particularly for building databases, we need to look things not published in English. There are many treasures in all different kinds of languages. So with that in mind, when I was a PhD student many years ago, I created this South American Mycorrhizal Research Network. I was telling Zoe right before this that when we launched this website in 2017, like 3 months later, I received an email by Diana Wall, and that was wonderful because we were brand new. So she was very aware of all of this, and it was a huge motivation right at the beginning. So this network is almost 600 people. We have a YouTube channel with more than 60 interviews to world-class mycorrhizologists, but also to early career researchers, and we have produced Springer books and scientific articles, and anyway, we collaborate with all this ecosystem of societies interested in societies and networks and NGOs interested in soil biodiversity. We have organized 3 symposia so far. The next one is in Quito, in Ecuador, this year, in August. And that's the YouTube channel, which I invite you to check. You can also easily join here. And I think that the next step on how we are doing soil biodiversity research from the Global South is to include everyone, not to keep things for our academic world, but to include everyone. I think this is one of the first steps, this study with the Atacameño people in the Atacama Desert. This study is ongoing, but they selected the sites of sampling, which is wonderful. So I think that's one of the solutions. And so I conclude by saying a little bit about the next steps. I explained you before what the units of selection means in mycorrhizas, but I think the next important step is to understand mycorrhizal traits. There is a huge adaptationist bias in how we do trait ecology. Not all traits contribute to fitness. Not all traits will help an organism to survive more. So we need to keep that in mind. We need to differentiate the type of traits and adaptations that we are investigating. And of course, I recognize that multi-level selection is one of the different ways that symbiosis, that cooperation occurs in a symbiosis. At finer or smaller scales, I think that there are other mechanisms going on, particularly direct reciprocity, which has been very well investigated and has very solid research. But I think that we need to put focus on what scale are we studying. And of course, there are also frameworks in plant soil feedbacks and complexity science tools and so on that need to be integrated. So as conclusion, mycorrhizal functioning is context dependent. Mutualism increases under limited and stressful conditions. There is ample and abundant evidence of multi-level selection across many different taxa and in situ and through experiments. And this framework that Nancy Johnson and I are proposing, functional team selection, predicts that under those limited or stressful conditions, below ground microbiomes contribute to positive plant soil feedback, increase host fitness and growth, depending on a selective force, host phenotypic constitution, microbial diversity, and time. With that, an invitation to this brand new journal. I cannot help but do this in this kind of audience. It's an NPJ Soil Ecology, just launching, and there are call for papers there. And here is my contact information, and thank you so much.